Now it should be fine.
Well, meanwhile,
Phil, do you want to pop yours up really quick?
Yeah, no problem.
Awesome, thank you.
And greetings to everyone coming on. Give us just a minute or two for the AV technicalities.
We got you, Phil, looking good.
All right, thank you.
All right, we don't remember, was there an order here? Phil or Judith, are you, either one of you expecting to go first?
I think I was first, but I don't mind going second.
Well, no, then that's perfectly okay. Is that okay with you, Phil?
Yeah, ladies first.
Ladies first. That's an outdated concept, but okay. All right.
Chivalry still exists.
We can say it's because of my family name that it's an A.
It's alphabetical.
Exactly.
Okay. So we'll go ahead and get started. It's a little after 2. Welcome everyone to another Pathways seminar opportunity. We have 2 presenters today, both who received Initiator Awards, our Career Starter Awards. And both have done, both of our speakers today have done really well. Our first speaker is going to be Judith Agudo from the Dana-Farber Cancer Institute. And her presentation is Harnessing Immune Privilege Mechanisms from Stem Cells to Protect Beta Cells from Immune Attack. And let me just say that both Judith and Phil presented at our Pathways Symposium at ADA, but they have both promised me that they have some new and additional material to what they presented then. So we didn't change the order, and we're looking forward to learning even more from Judith and Phil. Judith, please.
Awesome. Yeah, so I will go into more detail because I felt like everything was so broad when I presented
Right.
at the same time. So I will still, is it okay now? Can you see my full screen now? Not yet? Oh. Sorry.
Still not full screen, Judith, but we can see it okay. So I'd suggest you go ahead.
Okay, then I just keep going like this. Maybe let me do this. I'm sorry. I don't know what's wrong with my computer. Yes, so you will, I mean, I'm gonna, how do you say, spill the beans with my slides ahead, but it's okay. So I will try to go very quickly on the stuff that I already presented, but I feel like I need to re-explain it to make sense of what we are doing in the lab right now. I just want to explain that I actually, even though I'm supposedly an immunologist right now, I have been working in diabetes for like over 15 years now. I started in beta cell regeneration back in my PhD when I was still in Barcelona, and I was regrowing beta cells, which it works in mice. Actually, Andy Stewart here, he's an expert in that. I was reading a lot of his papers back then. But something that was very frustrating back then was that you regrow these beta cells, and this is great for type 2 diabetes, but then for type 1, I don't have to explain this to this audience. Then if you manage to replace those missing beta cells, the immune system is gonna go back and kill these beta cells. And I mean, this is because of a complex immune response that actually back then I did not understand at all. And with this idea in mind, once I finished my PhD, I got Fulbright postdoctoral fellowship, and I went to the Immunology Institute at Mount Sinai to learn immunology. The first part of my postdoctoral training was to develop tolerogenic vaccines to stop type 1 diabetes, targeting dendritic cells. And during that time, I had a very naive question, which I used to discuss with my mentor, Brian Brown, which is, I mean, NOD mice, right? Like we have type 1 diabetes in mice, in people, right, like across species, and it's always beta cells that get attacked. There's no such a thing as an alpha cell autoimmune disease happening, not in mice, not in humans. So we do understand that there are haplotypes that make a person susceptible to type 1 diabetes, but then why is it that it's always beta cells? And I felt, I did not have a proof, but I felt that beta cells were very sensitive, if that makes sense. And we didn't have a way to study that, right? Like we had the feeling that that cells in the body have different susceptibilities to immune attack. Some were more prone to die, others were more resistant, but there was no way for us to test. And then we, as immunologists, we decided, well, maybe we can develop a tool to study such a thing. And the way immunologists study immune responses is by using what it's called TCR transgenic mice, so T cell receptor transgenic mice. These transgenic mice, they have a T cell receptor that recognizes a model antigen. So for example, if I want to study immune responses against beta cells, I can use TCRs that recognize insulin. And if I want to study an immune response against alpha cells, I could use TCRs against glucagon. And if I want to study how, right, like various cells in the body have different susceptibilities, ideally you need to make a TCR transgenic mouse for every single cell that you want to study. That was obviously not feasible. And then Brian, my mentor, who's very clever, had this idea, which is, well, there are lots of mice that express GFP in many different cells. Why don't we use GFP as a model antigen? And all we have to do is to generate a TCR that recognizes GFP. And then you can study, right? Like if you have a mouse that expresses GFP in beta cells, you can study that in alpha cells, in brain, whatever. And that's basically what I did. I vaccinated mice against GFP. I generated isolated T cells that were specific for GFP. And since these mice so we use these T cells to develop mice. And since these mice were just GFP death-inducing T cells, we called, you cannot see it now, but the mouse were named Jedi because they recognize GFP, so we call the mice Jedi. And what did we do with these T cells? Well, the first thing I did was what I knew best, which was to study type 1 diabetes, or to study diabetes in this case. So one cell type that I knew was susceptible to T cell attack were beta cells. And luckily for me, there was an mouse insulin promoter transgenic mouse that expressed GFP available in Jackson Labs. And I got these mice. And it was as easy as getting the mice, adaptively transferring these Jedi T cells. And what happens, it's too bad that it doesn't, let me see. Does it work now? No, right? Why?
Still not full screen, sorry.
Yeah, it's because it's not gonna, anyway. Sorry, huh? And I did try, but I guess with the Zoom. Okay. So when I adaptively transferred these activated Jedi T cells in mice that express GFP in beta cells, something that happened was that these mice developed hyperglycemia, as you would expect. And then, of course, when I went back and I looked at the pancreas, the beta cells were gone. One very nice advantage of having GFP after a long PhD doing immunohistochemistry to see insulin was that with GFP, you don't need any staining whatsoever. You just get a section and you see GFP. So I could very easily see beta cells. Another advantage, which I could see with insulin, but I was not so aware until I had GFP, was that, for example, this one beta cell here has GFP. So I know has the antigen and it has a lot of T cells around it. So these T cells recognize GFP because I know these are Jedi T cells, and yet the cell is alive. So now, since this is GFP and it doesn't need any staining, I can get this beta cell back and see how come this guy is alive, even though it's surrounded by T cells. And this is something that our lab is very excited now to work on. So we published this work. And once we had this tool, other questions we had was, okay, so what else can we learn? Of course, beta cells are susceptible. Many other cells were susceptible. Can we find something that is not susceptible to T cell killing? So we then turned our attention to stem cells, because in the literature, you can very frequently read that stem cells are immune privileged. But then when you go and look for the actual experiments, you realize that there was no formal investigation showing that indeed stem cells are immune privileged. And I wanted to find a source that would inspire my research in, right, like how I can protect cells from autoimmunity, beta cells and other cells. So are really stem cells immune privileged? And this is a very important question, because if a stem cell is immune privileged, when that organ gets attacked, if you stop the attack, that organ can regrow. Conversely, if they are not, then the whole organ, right, like is doomed to die, and you need something else to regrow that organ. And to answer whether or not tissue stem cells, the ones that would really be susceptible to autoimmunity, were susceptible, or conversely, they were able to resist T-cell attack, I got these mice that were developed by Hans Clevers in the Netherlands that express GFP under the control of the LGR5 gene. And these mice have GFP only in stem cells, and actually in several types of stem cells at the same time. So in the intestine, in the ovary, and in the hair follicle. And I will show this very quickly. So first, in the intestine, we thought that this would maybe be a cell that is immune privileged, because the intestine is obviously a very important organ. And for those that are not used to flow cytometry data, so GFP, this is GFP intensity, so these are the stem cells. And when you inject Jedi T cells into these mice, all the stem cells are gone, so they die. What happens with the ovary? Well, the same thing. These are in the untreated mice, and once you adoptively transfer Jedi, they are all gone, so they are not immune privileged either. What about in the skin? There are skin stem cells that have GFP in these mice. What happens with them? And to our very big surprise, these are the GFP cells in the untreated, and these are the GFP cells in the Jedi treated mice. So they were 100% resistant, not a little bit, 100%. And many iterations, how is this happening? Well, it was not lack of recruitment. The Jedi T cells were making it to the skin. The skin is not an immune privileged organ. There are actually lots of autoimmune diseases that happen in the skin. How could this be happening? And very, very long story short, what I found was that these stem cells in the skin, in the epidermis, they actually had very low expression of class 1. This is class 1 in the immune cells in the skin, class 1 in mature keratinocytes, and these were the stem cells. And MHC class 1 is actually the receptor that binds the T cell receptor on the T cell. So this is what T cells see. A CD8 T cell has a TCR that binds to class 1. This is how the T cell knows this is the target that has to kill, and then decides to kill. So if you don't have MHC class 1 or not enough, you become virtually invisible to T cells. And how are these stem cells downregulating class 1? Again, very, very long story short, finally, I decided to do RNA sequencing, which is, I guess, the blunt way many people try to figure out mechanism when you really don't know where to go. And I isolated these stem cells from the skin and cells that were very closely related, but were susceptible to T cell killing. And the profiling of these cells, what revealed was that, and this was really nice. So these are the genes for class 1, H2K1 and H2B1. They were highly downregulated, but this was beyond the expression of MHC class 1. These stem cells had low beta-2 microglobulin, which is the co-receptor, low expression of components of the immune proteasome, which is what allows here, this is the immune proteasome, it allows the cleavage of proteins to obtain the peptides that are loaded on class 1, low expression of TAP1 and TAP2, which are the molecules that allow this loading or this entrance of peptides into the ER for loading on class 1. So the whole machinery was low. So these cells really wanted to not present enough antigen to T cells. And what this resulted on, I think it's like really, really cool, is that these cells are not really class 1 negative. They still have some molecules of MHC class 1, but because the whole machinery is so low, every single step is very inefficient. And T cells for killing, they need class 1, but they need class 1 with the right peptide, right? So if this TCR is recognizing, let's say, insulin, if you have another MHC here that is presenting, I don't know, GAD65, this one is not going to bind here. It's only going to bind with the one that has insulin. So if you have the whole machinery down, you are not going to have a critical mass of receptors or class 1 with the right peptide for the T cells to bind. And what this results to is that these stem cells in the skin, since they still have some molecules of class 1, they are completely resistant to killing by NK cells, by natural killer cells. And natural killer cells, what they do is they kill cells that are deficient in class 1. So if you want to protect the cell by knocking out MHC class 1, or usually B2M, beta-2 microglobulin, those cells will get attacked by NK cells. But the stem cells are not, because they still have a little bit of class 1. They just bring everything down. So what is upstream, right? Like what is allowing this whole complex program that everything is down? And again, another very long story short, we found that this gene NLRC5 is a master regulator of the whole antigen presentation pathway, is actually missing in the stem cells. And I was able to prove that when you put this gene back in the stem cells, then you recover antigen presentation and they become susceptible. So NLRC5 was the gene. So when I moved here to Boston and I started my lab, I was like, well, now it's my chance. I'm gonna take NLRC5 and I'm gonna use it, right? Like no one thought of this gene, right? Like we think about B2M, you remove B2M, you add PBL1, and then you put something to stop NK cells. So what, instead of having this cumbersome combination of genes, why don't we use what, for example, these fully immune privileged stem cells that even if you throw at them millions of activated T cells, they don't get killed and they also don't get killed by NK cells. And everything is because of this one gene. Can we actually use this knowledge and just knock out this gene in beta cells? And this was my idea. That because NLRC5, as I said, controls many of the steps. And when it's missing, it doesn't bring everything to zero. It just, in a combination, right, like altogether goes to a low level. And it's actually something else that is great about NLRC5 is that this is the sensor that responds to interferon gamma. So when a cell senses interferon gamma, one of the things that the cell does is to induce class 1. And that makes the cell more susceptible to T cell killing. That upregulation of MHC class 1 is mediated by NLRC5. Right, like interferon gamma is sensed and induces expression of NLRC5. So if you block this, the cell is no longer to upregulate class 1 in response to interferon gamma. And this is basically the idea I brought to ADA, and it got funded. So it was really, really, I mean, it was really amazing. It allowed me to go back to diabetes, even though I'm in a cancer institute. The project started in February 2020. So I don't have to tell you what happened in February 2020. When the lab was shut down, we looked in the literature for cues or clues that would enforce our, our rationale. And for example, this is microarray data from this paper, and we downloaded the data, and you can see these are isolated islets from NOD mice as they develop diabetes at day 0, 3, 4, and 8. And you can see very nicely how NLRC5 is induced during type 1 diabetes. Make it, and I think this makes a compelling case for, well, you want to stop this from happening. This is data from patients. And this is something also that I have learned during this last year with work in my lab, is that actually, after having studied MHC class 1 in so many organs, it's very interesting. Beta cells actually have very low MHC class 1, comparable to neurons. But when there is an immune attack, right, like when there are lots of immune cells infiltrating, these are pancreas from patients, you can see that class 1 gets massively upregulated. And this massive upregulation is what allows now T cells to engage and kill. Now in my own lab, we have recapitulated this, and you can see very nicely the pancreas has very low class 1, and this is with our Jedi T cells, but the same thing happens with NOD mice. When there is immune attack, class 1 goes up, and this is what allows the killing. So what we have to do is prevent that striking I was about to say abnormal. It's not abnormal, but it's really bad for the beta cells. This huge increase in class 1. So we started to knock out NLRC5. We have been working with primary beta cells, and we have also been using NIT1, which is a cell line, and I absolutely do not like it because having worked many years in my PhD with true beta cells, a cell line doesn't, fulfill me, but it also has been helpful as an easy readout to test the gene. And all this is, of course, work in progress, but I can, I'm very proud to show to this audience that, for example, in this NIT1 cell line with GFP, we put these GFP NIT1 cells in mice, and then we inject Judith T cells. So the Judith T cells recognize GFP and they kill the cells. When you knock out NLRC5, we get a very nice protection, even better than B2M knockout alone. And we think it's because we are protecting from T cells, but at the same time, we are protecting from NK cells, or at least we are not triggering an undesired bystander killing by NK cells. The other aspect here that I want just very quickly to highlight is that someone may think, well, if the problem is interferon gamma, maybe it's just easier to knock out the interferon gamma receptor. And then you don't upregulate class 1, you don't upregulate anything else. Interferon gamma receptor has some upsides, I guess you can say, some benefits for the cell, because, for example, a very famous gene now here with my coworkers in the Cancer Institute is PVL1. So PVL1 is induced by interferon gamma. If you knock out the receptor, you no longer induce PVL1. And we believe that by knocking out NLRC5, you block that induction of class 1, which is the thing that you want to block, whereas you are still preserving the upregulation of PVL1. And I think this was, yes. And in the meantime, we are working on NLRC5. This is just one candidate, and I do not want to commit my whole career and try to be able to successfully transplant islets with just one gene. So we are actually, we are still studying these immune privileged stem cells because they really have a lot of things to teach us. They do lots of things. They recruit Tregs. Different types of stem cells recruit Tregs in a way that we don't understand. So if we were able to learn that, we could engineer beta cells to recruit their own Tregs. And then also, I'm very, very excited to study these cells that I was showing before that I found during my postdoc. Now that I have my own lab, we are trying to isolate these GFP beta cells here and learning how are they able to survive in this harsh environment. Because the GFP here is under the insulin promoter. So this means that this cell is producing insulin. It's not that they are differentiating or losing, maybe to some extent, but at least they are able to produce insulin. So whatever they do, I think we should learn and try to exploit. And with that, I don't want to go over time. Obviously, I want to thank my previous mentors, my PhD mentor who taught me everything I know about the pancreas, and my postdoctoral mentor, Brian Brown, and Miriam, who also taught me a lot of about immunology, the funding I had during my postdoc. And then in my own lab, I have, as a postdoc that just recently started 2 months ago, everything got delayed because he's under a visa, and a very good technician who has been doing the NLRC5 knockout work. Of course, thank you first of all, thank you a lot to Dr. Stewart, to Andy. He has been a tremendous mentor. As soon as I got the award, I sent him an email. He sent me back his answer right away. We had several meetings. He even assembled a team of people to make sure that I had all the mentoring I needed and I could answer all the questions I had. He was extremely generous with his time. I want to thank, obviously, the Pathway to Stop Diabetes Award, who has allowed me to go back to diabetes. I'm really thankful for that because this is my passion and the thing I know the most, if anything. And I also want to thank Doug Melton because thanks to this award, actually, I went back to, or I was able to study diabetes to a much larger extent than what I would have been able to here. And when I was presenting some of my ideas, Doug saw me and he loved what I was, that's what he says. And he has become another mentor to me. And thanks to him, I'm now part of this JDRF New England Center of Excellence, where we have very exciting discussions of how we can get stem cell derived beta cells in this case. That's what he's obviously generating, how we can make them invisible to the immune system. And yeah, that's it. Thank you. And thank you so much, everyone, for your attention.
Wonderful, Judith. Thank you so much. So a couple questions have come in via the chat. First, Rohit Kulkarni, great talk. Are the resistant GFP positive beta cells dedifferentiated? And do they express markers of mature beta cells?
We don't know yet. That's something actually we had discussed with Andy in the past. Yeah, all I know so far is that they express insulin. Even with Andy, we had been discussed of like some lineage tracing because they could be coming from alpha cells, like when they show up, like, yes, EDU experiments to see probably all that is just ideas that we have to do. So I don't know yet. But yeah, that was a fear I had, that maybe what allows them to escape. The one thing we have seen is that they are class 1 low. So these beta cells that don't get killed by Jedi T cells somehow are able to not induce class 1 naturally by themselves. So okay. I think it has to do more with that than with redifferentiation.
Okay, Marlon Pragnell, really nice talk. Have you measured NLRC5 in NPOD pancreatic samples? So switching to human pancreas sections, yeah.
Yeah. So we did actually during the lockdown, yes, and it's high in the patient, like it's high when there is, and it actually in all the autoimmune diseases, NLRC5 is always high.
Right. Okay, and I think you said entirely too many nice things to Andy Stewart. I saw his head actually swelling on the screen. But Andy has a question. What are the cells you silenced NLRC5 in? Which cells did the silencing occur in?
The silencing naturally, you mean, or that?
No, sorry. You showed us a cell line that you silenced it, but yes. I didn't catch the name of the cell line.
NIT1. It's a cell line. It's an insulinoma from NOD. So it's not a beta cell.
Hamster line, right?
Sorry?
I think it's a hamster line, right?
No, it's NOD.
Oh, that's right. NOD. Okay, got it.
Yeah. So we are now trying to engineer beta cells. So now it's when I will have to knock on your door again, because engineering beta cells is much more difficult than a cell line, obviously.
Well, apparently there are people in Boston who do that, I understand.
Wow.
So another question that Andy asked, what are the next steps in this story? And I want to elaborate on that too. As you dream about how you're going to translate this information to human type 1 diabetes, there's some tough obstacles. There's the window of time you need to detect who is going to go through to actually get an autoimmune attack and beta cell destruction. So you need to know which patients to apply your intervention in. There needs to be some beta cell specificity for immune response elements that are actually broadly dispersed in the body. So what's your thinking about how this can actually be translated? Just generally.
I mean, like just dreaming, I think you should be able to apply these to patients regardless of how long they have been diabetic or they have had the disease.
Okay.
Because if you manage to engineer beta cells, or for example, Doug, what he wants to do is to take stem cell derived beta cells and engineer them and then put them back. They should be able to escape at least from the autoimmune attack based on our data. A different story is the allograft rejection.
Yeah.
Right.
Yeah.
That's the other part that we are now testing, because class 1 is so low that I think it can work for allograft, but that's what we have to test, because otherwise you're very limited, right? Like you need to be able to survive the allograft. That's the major limitation here.
Okay, great. And last question from Tony Ferrante. does NLRC5 deficiency increase the ability of the cells to avoid anti-tumor immune targeting?
Yes.
Yeah,
NLRC5 is one of the genes that shows up in many CRISPR screenings. It's just one gene that people don't pay attention to. But it's one of the top always. Yeah.
Okay, terrific. All right, Judith, thank you so much. That was really clear. And I thought it really did give us another perspective beyond your ADA talk. So I appreciate you bringing a broader array of information in. That was really well done. Okay, with that, we'll move on to the next presentation. Phil, if you could be getting your presentation up. Yep, start sharing screen. Okay, the presenter here is very well known to me. Just down the hall, my faculty colleague at the Duke Molecular Physiology Institute. Phil, like Judith, is a winner of an Initiator Award in 2016. And you can see the title, The Protein Phosphatase M1K, a new player in beta cell function. Phil. Are you on mute?
Now I'm off mute. Sorry about that. So like Judith, I'm going to delve a little deeper into one specific story that was kind of covered briefly in my Pathways Award. So I hope, in my Pathways seminar. So I hope this is going to be interesting to everybody. And we've been focusing a large part of the Pathways, my Pathways Award, on this novel protein phosphatase, PPM1K. And the reason we became interested in this phosphatase, as it has been described to be the phosphatase that dephosphorylates and activates one of the rate-limiting steps in branched-chain amino acid metabolism. And as everyone will be well aware, is that elevations in branched-chain amino acids are highly associated with insulin resistance and future development of type 2 diabetes. And so we became interested in this particular regulatory node, which is made up of the phosphatase PPM1K and its reciprocal kinase BDK, and how they may play a role in controlling metabolic dysregulation on the path to type 2 diabetes. And our initial focus was within the liver because the balance of, or the equilibrium of these two enzymes is disturbed with obesity, such that there's more kinase and less phosphatase, leading hyperphosphorylation of this enzyme BCKDH, and inhibition leading to accumulation of the branched-chain keto acids and the branched-chain amino acids. And we first started exploring what would happen if we reactivate this node, either by overexpressing the phosphatase or inhibiting the kinase. But the phosphatase is also interesting in the sense that not only does it regulate this key step, it also came out through genetic studies as being the one loci that's associated with elevations in all three branched-chain amino acids. Here's isoleucine, leucine, and valine, and can also be directly associated, SNPs upstream of this gene, with risk for type 2 diabetes. And so we can see the p-values here for diabetes effect. And so PPM1K is both associated with branched-chain amino acid levels in human populations and risk for type 2 diabetes, and it controls this regulatory step. Looking at some of the first work we did where we tried to regulate or restore levels of PPM1K in the liver, we did that using adenovirus overexpressing PPM1K, and the results were quite striking. And here is the adenovirus on the bottom, comparing to the effects of inactivating the kinase at the top. And you'll see both interventions lowered branched-chain amino acids as would be expected, lowered the direct intermediate that's consumed by BCKDH, the enzyme that they regulate, more substantially. And there were these salutary effects on hepatic lipid content, as well as glucose tolerance after only 1 week of treatment with these interventions in fatty Zucker rats. And this was pretty astounding and resulted in a publication describing a new role for both the phosphatase and kinase in regulating some other enzymes within the liver. And so the model that we developed from this was that both the kinase and phosphatase don't only regulate BCKDH, this enzyme in branched-chain amino acid metabolism, but they also regulate the phosphorylation of other enzymes. And the one we focused on was ATP citrate lyase in liver, which is a key enzyme in de novo lipogenesis. And as part of this work, we were able to show that both the kinase and phosphatase were sensitive to glucose exposure and fructose exposure, such that activation of CHRBP induced the kinase and suppressed the phosphatase. And that altering the balance of these enzymes could ultimately alter the phosphorylation of ATP citrate lyase, resulting in shifts in rates of de novo lipogenesis that we attributed to some of the beneficial effects related to modulation of this node. And so this is all well and good, and we felt that that could potentially explain these associations between PPM1K and diabetes risk. However, about the same time that we published this, some other studies came out looking at genetic associations between PPM1K, branched-chain amino acids, and insulin resistance to type 2 diabetes. And what these studies reported were that although the associations between SNPs and PPM1K are present for type 2 diabetes and branched-chain amino acids, they're not present with insulin resistance, suggesting that the presence of these SNPs in the context of insulin resistance promotes future development of type 2 diabetes. Another model that we felt could explain this might also be that PPM1K might be having direct effects at other sites, such as the beta cell, which hadn't been explored, and that could be promoting progression to type 2 diabetes. And this could either be via effects on branched-chain amino acid metabolism within the islet, or the effects of systemic changes in branched-chain amino acid and their influence on the islet, or through other targets, such as those that we had identified through our phosphoproteomic screen in the liver. Additional support for a role of PPM1K in the islet came out of this unbiased systems genetics approach study that was conducted by Leif Group's team, where they looked at genes that co-localize with genes around diabetes SNPs in human islets. And they actually found that PPM1K was one of 12 genes that was associated or correlated tightly with at least 4 type 2 diabetes CETA genes. And if they looked at levels of PPM1K expression within human islets, and I think this is from 51 different individuals, it did indeed track with both insulin secretion, these dots in red. So higher PPM1K expression is associated with better insulin secretion in islets, and also glucose control as measured by HbA1c. So there seems to be some support that PPM1K might have a functional effect or be involved in heretofore undescribed functions within the islet. But no other work has been done on PPM1K in this realm, and there's really been no direct test about what modulation of PPM1K does within the islet. So Jan Delay, a talented French postdoc in my group, has really taken this project by the horns and has been exploring what role PPM1K has within islets. And so this first slide is to simply show that PPM1K is actually well expressed in islets. And if you looked in the GTEx database, you actually see that whole pancreas levels of PPM1K, the expression looks relatively low. It's definitely not among the most highly expressing tissues for PPM1K expression, but a new database looking, which called TIGER, which compares islet gene expression with that already in GTEx, shows actually PPM1K expression in islet is among the most highly expressed levels across the whole organism, suggesting it's probably quite relevant within the cells of the islet. And our data shows that in the setting of type 2 diabetes, there's actually lower expression of PPM1K within human islets. There's a nice single cell RNA sequencing data available in the Power Brisova lab. And we can see that within human islets, PPM1K is well expressed within the alpha and beta cells, also the delta and gamma cells somewhat, suggesting that PPM1K is expressed in relevant cell types for progression to type 2 diabetes. And so Jan's work starting in INS1 832.13 cells shows that when he knocks down PPM1K using siRNA, There are effects on beta cell proliferation. At 72 hours is quite a reduction in proliferation. And this occurs concomitant with an increase in glucose-stimulating insulin secretion. And these data are mean plus the standard deviation. We see there's a significant increase in glucose-stimulating insulin secretion at 72 hours after PPM1K knockdown, despite there being a reduction in proliferation. These effects can be recapitulated in primary rat islets. Although the knockdown was less successful there, we see the same trend for reduction in proliferation, as well as a significant increase in glucose-stimulating insulin secretion. Looking at gene expression profiles, there's also very interesting effects on an array of genes. I'm just showing a few interesting examples here that are related to beta cell identity. And here we see MAF alpha and NKX6.1 have reduced expression, while genes typically associated with alpha cell phenotypes, ARCs and glucagon, are actually induced. And then CHLBP beta, which I spoke about earlier as being a transcription factor that represses PPM1K expression, it seems that there's an interplay between these two, as when we knock down PPM1K in beta cells, CHLBP beta expression is remarkably reduced. So how is this happening, and is it through the known pathway? In fact, when Jan went ahead and looked at the effect of PPM1K knockdown, here's the protein levels on the known target BCKDH, we don't actually see an increase in phosphorylation on the predicted target site. If anything, there's a slight reduction in BCKDH phosphorylation. It turns out that when you knock down PPM1K in INT1 cells, there's a reduction in the expression of the kinase, and we think that may account for this slight reduction in phosphorylation. And our stable isotope tracing in these cells is supportive of the fact that knockdown of PPM1K does not inhibit BCKDH activity. In fact, if anything, there's a modest trend for increased labeling of a downstream intermediate 3-HIB. All of this to say, although knockdown of PPM1K influences an array of beta cell phenotypes, it doesn't seem to be due to inhibition of branched chain keto acid dehydrogenase activity. This seems somewhat surprising, but if we revisit the paper that identified PPM1K as the essential BCKDH phosphatase, in their whole animal knockouts, they see an effect to increase phosphorylation on this known site, serine 293 of BCKDH in liver. However, in heart, there is absolutely no effect of PPM1K knockout on 293 phosphorylation, suggesting that this is a tissue-specific effect. And so our take on this at the moment is that PPM1K is not a dominant player in the regulation of BCKDH in the beta cell. So could a novel function of PPM1K underlie the effects we're seeing on beta cell physiology? And so we revisited our phosphoproteomics data that we published in liver. And in addition to ATP citrate lyase phosphorylation, overexpression of PPM1K led to reductions in the phosphorylation of the major regulatory phosphosites in ribosomal protein S6. And this was striking to us. We noted that these phosphosites contained the phosphorylation motif that we identified for PPM1K. And we also noted that there was no change in mTOR or X6 kinase phosphorylation in this data set, suggesting that PPM1K might be the S6 phosphatase. And to the best of our knowledge, there's no other described phosphatase for S6. So Jan went ahead to explore this, and this was the model. One explanation for how PPM1K could be affecting S6 phosphorylation could be through its effect on BCKDH. And of course, our phosphoproteomics data came from liver, where we did see an effect to reduce phosphorylation of BCKDH. And the idea here would be that that would lead to increased oxidation of branched chain amino acids, resulting in reduced inhibition of the cestrins, leading to inhibition of mTOR C1 and S6 kinase upstream, and then reduced S6 phosphorylation. Although we were interested in whether PPM1K might also be directly dephosphorylating or regulating S6 independent of mTOR C1 S6 kinase, since our phosphoproteomics data did not show changes in the phosphorylation state of these enzymes. So Jan went ahead and looked at this signaling in the INS1 832.13 cells. Here you can see we've knocked down PPM1K in the right two lanes. This did not result in any changes in the phosphorylation of mTOR or P70S6 kinase. However, we did see robust increases in the phosphorylation of the regulatory sites of S6 that we identified in our phosphoproteomics, suggesting that PPM1K does indeed regulate S6 phosphorylations and in the INS1 cells. Importantly, looking at other potential regulators of S6 phosphorylation upstream outside of the mTOR pathway, being the ERK and P90 RISC pathway, we didn't see that knockdown of PPM1K regulated either of these pathways, which suggests that PPM1K is likely regulating S6 directly. Importantly, these changes in phosphorylation of S6 relate to effects on protein translation. This is using pure myosin incorporation in a pulse chase experiment to gauge rates of protein translation within the INS1 832.13 cells. And here you can see compared to control SI treated cells, those treated with the PPM1K siRNA have a robust increase in pure myosin incorporation, suggesting a greater than 1.5-fold increase in protein translation within those cells. Doing polysome profiling shows us an increase in polysome formation. And these cells actually have a greater protein content per cell. So PPM1K knockdown results in increased S6 phosphorylation without changing mTOR or S6 kinase upstream. And this relates to an increase in protein translation and increased protein content within those cells. Increased protein translation also seems to result in the activation of an unfolded protein response. And here we have some hallmarks of that with increased XBP1 splicing in PPM1K knockdown cells, as well as increased expression of BIP, two markers of the initiation of ER stress response. And we think this could be an important component of the connection between PPM1K and type 2 diabetes, as increase in ER stress response within beta cells has been implicated in eventual beta cell failure and death. So Jan wanted to go back and be thorough and demonstrate that these effects of PPM1K are not related to the regulation of BCKDH. So the experiment that he devised to do this was to activate BCKDH with the inhibitor of the kinase and see whether that was able to reverse the effect of PPM1K knockdown. And so here you can see in the control cells that knockdown of PPM1K leads to the increase in phosphorylation of S6 without changes in mTOR phosphorylation or S6 kinase phosphorylation. If you treat with BT2, you lower the phosphorylation of BCKDH. However, this has no effect on downstream phosphorylation of S6, which remains increased when you knock down PPM1K, suggesting this is not related to regulation of BCKDH. Then Jan wanted to revisit whether PPM1K really is uncoupling S6 phosphorylation from upstream signaling, as was suggested by his Western blot. The way he went about this is to look and see whether he could disable mTOR signaling and see whether there was a maintenance of higher phosphorylation of S6. So he did this at first by doing a time course of rapamycin treatment. Of course, rapamycin will inhibit mTOR C1 activity. And what we have is the control SI treated cells versus the PPM1K treated cells on the right. What we see is that rapamycin treatment rapidly reduces phosphorylation of P70 downstream of mTOR C1, equally across both groups and throughout the time course of the experiment. However, the increase in phosphorylation of S6 is maintained throughout the experiment in the PPM1K knockdown cells, suggesting that PPM1K is exerting this effect to increase S6 phosphorylation independently of mTOR signaling through P70, S6 kinase. Another way to do this, which we're able to sustain for longer, is activation of AMPK, which will inhibit mTOR, and we can take this out to 12 hours. And so here we see ICAR increase the phosphorylation of AMPK as expected, equally across both groups. This led to an equivalent reduction in P70S6 kinase phosphorylation. However, even out to 12 hours, PPL1K knockdown cells maintained a higher level of phosphorylation in S6. Jan's very thorough, so he wanted to take another method to do this. And in this case, he tried to inhibit PI3-AKT signaling with workmenin. This was successful, and you can see that there's a reduction in AKT phosphorylation with workmenin treatment. As reported before, the effect on phosphorylation of S6 is maintained throughout. However, the really interesting finding that came out of this experiment was not that we could uncouple S6 from mTOR S6 kinase, was that it appeared that baseline phosphorylation of AKT was actually reduced in PPL1K knockdown cells. And this led us to wonder, does PPL1K knockdown produce a state of insulin resistance in the beta cells? And this might explain some of the phenotypic changes, at least at the transcription level, that we had observed. So Jan went ahead and treated the PPL1K knockdown cells or control cells with IGF and looked at signaling through the insulin signaling pathway. And what I hope you can see here is that knockdown of PPL1K does not change signaling through the IRS. However, at the level of AKT, there is reduced basal and insulin IGF-stimulated phosphorylation of AKT on both the threonine 308 and serine 473 sites. And he determined that this was associated with an induction of PTEN. And of course, PTEN will reduce levels of PIP3, which could explain the change in AKT without any changes up in the IRS1 upstream. And we see PTEN is induced not only at a protein level, but also at the mRNA level. Importantly, if you use a PTEN inhibitor, BPV pick here, we can see that we can recover insulin resistance in the PPL1K knockdown cells at the level of AKT. And so these works have led to a new model of a role of PPL1K in the beta cell. We think that PPL1K is an important regulator of S6, phosphorylation and protein translation, and then when you lose PPL1K either through effects of genetic variants or through changes in nutrition and et cetera, that you lose the ability to have temporal changes in protein translation. You have unmitigated high levels of protein translation, which increase insulin production and insulin secretion in the short term, but may make you more susceptible to beta cell failure through mechanisms related to ER stress, as reported by markers of ER stress that we showed, increased levels of XBP1 splicing and BIP. And we also think that this might lead to a feedback mechanism where PTEN is induced as a mechanism to reduce AKT signaling to mTOR, but an off-target effect of that is potentially the loss of beta cell identity through gene and transcription factors that are regulated by AKT. And so we are currently exploring this both in our newly developed conditional knockout PPL1K mice for beta cells, and we're also actively looking at this in human samples with anagloin to see if this model holds up. And I'll end there. And I'd like to thank large part of this work is carried out completely by Jan DeLea, postdoc in our group, but a number of other people in the group, including Jackie Waleko, have been involved in assisting him. Of course, wouldn't be here without mentorship from Chris Newgard and a lot of our collaborators, and of course, the support from the Pathways Award. Thank you.
Thanks very much, Phil. And let's see. There's a question here from several people. Okay. Stephanie Stanford. Great talk and interesting project. Is ppm1k localized in the mitochondria? If so, how does it access S6 as a substrate?
Yeah, so great question. ppm1k was thought to be mitochondrial or localized to the mitochondria. In our cell metabolism paper, we addressed that by cell fractionation and actually determined that although it is in the mitochondria, it's also found outside of the mitochondria as well. And there seems to be shifts in the cytosolic fraction that are responsive to feeding and fasting responses as well. So it was first reported there, but there's not been a lot of work done on it. And it turns out that it actually exists elsewhere as well. So we think it may be as likely direct.
Okay, thanks. Gene Schaefer, great talk. Do you think RPS6 phosphorylation changes ribosome function? Any predictions on how phosphorylation might impact the cryo-EM structure of the ribosome?
I can't speak to the cryo-EM structure of the ribosome. That's really out of my wheelhouse, but I'm going to have to talk to you more about that. We are planning ribosomal footprinting studies. So what we want to understand is, does it change the specific transcripts that are interacting with the ribosomes and the polysomes? And so we're developing that method in our lab at the moment. And so we're hoping to see whether we get shifts in the actual transcripts that the ribosomes are interacting with, and we think that might be important.
And then Alan Saltiel, beautiful talk. Is the specificity of ppm1k explained purely by its consensus phosphorylation site, or is there a scaffolding targeting protein? Alan is stealing from old work on glycogen targeting subunits here, which I happen to know a little bit about. So any idea of a scaffolding targeting protein involved in any of this? No
idea at all. There's very little work that actually been done on ppm1k. I think if you were to PubMed it right now, you'd see 30 papers talking about it, and half of those are reviews. And so we want to move ahead and do some studies where we're looking at interaction partners and what can we co-IP out with ppm1k, and also looking at the beta cell specific phosphoproteome. But yeah, there's really not a lot done there. So there's a lot of work that really does need to be done about how this is regulated. But I think if we can get to a point where we can think of strategies to either enhance interactions, interfere with interactions, or limit stability, or enhance stability, that this could be quite useful.
Right. Okay, and I mean, I think your talk shows that BDK is not the partner kinase to this particular activity of ppm1k. Is there a partner kinase for this pathway? And now that is the analog of BDK?
I guess one would think about would be P70 S6 kinase. And so for at least for S6, and so I think some of the work we need to do is look at how regulation of that pathway is influencing ppm1k levels.
Right.
One has done some work where we do see rapid turnover of adenoviral expressed ppm1k protein if we incubate cells with things that would stimulate mTOR signaling. And so we do think there's this kind of balance there where the cell, when it does have the right conditions to promote growth, will actually tune down ppm1k really rapidly. I think we had, within 5 minutes of adding serum, half of the overexpressed ppm1k protein was lost.
Right.
So that was pretty interesting, but we're still trying to figure out, what might be going on at this level.
All right, well, that was terrific, and it gets me really primed for the visit by Anna Galoyan to our institute in a month. I can definitely see why you want to go look at the genetics in more detail, especially in the islet. So I think with that, we'll bring it to a close. I want to thank both Phil and Judith for great talks. The next session is September 1st at 2 p.m., of course, and it will be Jonathan Flack and Ebony Carter. So until then, everyone, please stay safe, and we'll see you September 1. Thanks again. Bye-bye.